This is regarding the cellular mechanical strength and its flexibility. We know that, just recall the previous class, the mechanical strength of a biological cell, it is provided by these molecules, structural molecules. These molecules I call the microfilament, microtubule and intermediate filament. As you know, these are the molecules which can provide a mechanical strength, a tensile strength to the cell. And this is a molecule, intermediate filament, this is of interesting, which can provide, in addition to mechanical strength, it can provide a flexibility, that is plasticity, flexibility to the cell because of the content of the fiber, that is molecules or structures. It is fibrous protein molecules which are of this kind of arrangement or kind of this interaction. As you know, these fibrous proteins, finally what is that, twisting around each other, forming a units like this, single units. And further, these units are interacting with each other. It is both longitudinal and lateral. That is a staggered interaction. Staggered means a weaving interaction, weaving like interaction, just like the threads you are weaving into a rope. That is a kind of interaction. So these are weaving. I mean, these units now are weaving longitudinally as well as laterally. This kind of weaving or staggered interaction will induce this filamental structure. What is that? Finally, this filament, what we get now, a rope-like structure, induces a flexibility. That is, behave like a pakka flexible character. That is the one which is required. So, in spite of the tensile strength, it has a greater flexibility. So, the flexible property which is induced by what is that? This kind of structure. So, that is the one we needed, because in certain conditions the biological cells subjected to, that is subjected to twisting, the cells undergo twisting, folding and bending in certain cases. So this kind of what is that, mechanical stress what is applied on the cell, sometimes it will bend, it will be twisted, it will be folded, and after that action, what is that, it immediately regain the shape. That is most important. The regaining is important. That is the one, it is what is that, given by this kind of molecules. Flexibility, it will regain the shape because of the flexible character which is built inside the cell, cytoskeletal cell. So otherwise, if there is no kind of regaining or flexible character, the molecule thus bent or twisted will be what is that, broken, and finally it will collapse. That is the also what is that, that is the one I need to talk about more about the properties of these intermediate filaments. That is what kind of contribution these materials or structural materials give certain organs in cells, certain cells of the organs. So that I need to take up what is that, an example of organ is skin. Example is the skin I am taking to talk about more about the property of these protein molecules or structural protein molecules. As you know, what is the skin? Is the biggest organ, of course, is the largest organ of the body. There is the one that is a barrier between the surrounding and the body. That is important barrier. So let us look into the structure first, into the brief about the skin. What is the structure of the skin? The skin, what is that, is made of three layers, mainly three layers. First is, uppermost is epidermis, dermis and hypodermis. epidermis. I need to talk more about this. Epidermis, the detailed structures, again there are 5 different structures are there. So before going to this structural aspects of the epidermis, let me brief about, I don't want to go in details about this, what is the dermis layer and hypodermis layer. The first layer is epidermis, under which dermis, under which hypodermis. What is dermis? It is one which gives a connection, as act as a connector, connective tissue between the upper layer and the lower layer. Dermis is this one. Okay, that upper layer is epidermis, dermis, lower layer is hypodermis. So it contains many organs important that is required for the body. Organs are there. Hair follicle, glands are there. And before this, the tissue, what is that? This tissue is made of, what is that? This second layer, dermis, is made of the cells like fibroblast. This tissue consists of different cells. First of all, I go into the details about these functions. Let me brief about the cells of the layer. Fibroblast cells and immune cells, local, are residing here. That is not circulation through vascular system. It is local immune cells are there, and also some adipocytes cells are also there. Here the fibroblast is very important. It's a major cell content. The majority of them are fibroblasts because these are the one which synthesize most important protein molecules that is required for this tissue. What are them? Collagen and elastin. Collagen and elastin. So the collagen and elastin are extracellular matrix, are extracellular structures. Now actually I am talking about only intracellular structures. So later class, let us look into the extracellular structure. This fibroblasts synthesize the collagen and elastin and send out into the intercellular space as a extracellular matrix. That is the one what is that is required for this layer as to withstand the mechanical pressure. This layer should have some mechanical pressure as well as flexibility. The flexibility because of elastin protein molecules and mechanical, that is tensile strength, it is because of collagen material, collagen protein molecules. Collagens are as like fibrous proteins, but extracellular material, that is not intracellular because intracellular material. Yeah. So that is the one we need because this is what when you apply any pressure, because if you apply pressure now, it can withstand. That is the kind. That is why we need this. And below this layer, hypodermis, and you know that it is a layer containing what is a connective tissue as well as adipocytes. So mainly these are filled with what is that majority of them is adipocytes. Adipocytes are also there, very little, but adipocytes are the major content of the cellular content of this tissue. And also some immune cells. Immune cells are all over there. Here the immune cells are all there. Immune cells are localized immune cells, residing immune cells. So immune cells are there, different types of immune cells are there. So what is that? To act on many microbes, entry of microbes into the skin layer, accidental. That is not usually microbes will not enter because there is one not talking about the property of this. So that is immune cells are there. And just in brief about what is the function of this, that is called integumentary system, the A, and the glands are there. There are 3 different glands, that is sweat glands. There are 2 different sweat glands, eccrine and apocrine glands. Purpose is different. And one more is then sebaceous gland. This is hair follicle because skin is always covered. No, animals and usually human skin is covered with the hair. It's very important. That also play a biological role, functions to our body. Let me talk about the function, biological function of this hair and these glands. First, I will talk about the gland. This gland duct is drain into the hair follicle. What is that gland produces? This is cells are there. Usually, gland contains their own cells tissue. Here, gland. These cells produce oil. That is a mixture of lipid molecules. So, purpose of this oil is what is that? Through this will come out through the hair follicle to keep the upper layer which contains dead cells and also dry. Give them lubrication. What is that? To lubricate the cells on the upper layer and also the air on the skin. Lubrication because it is to keep it pliable. Otherwise, it becomes very dry. So that is the purpose of sebaceous gland. And the purpose of these air bumps, air and these gland. This is the gland actually you should know. It is because of the sweat and the salt, of course. More salt and sweat will come out. Only when there is a hormonal change. But in this case, this active hormonal change, but it drains into the hair follicle. So only during the hormonal change or during the nervousness, when the body becomes nervous, then you see the sweat coming out. That is, this is the apocrine sensor. Autosense is there and the sweat is coming out on the skin. That is the purpose. And this is important, acrine sweat, that is that is drained into the pore on the skin outer surface, directly into the surface. This is important. So this sweat gland and this integumentary system are the hairs and this sebaceous, what is that? Cell contained adipocyte. So a sheet of what is that? A sheet of what is the fat cells are there, very important. This and this and this are thermoregulatory. They regulate the temperature of the body. That is, keep the temperature almost constant. When they rise, usually there is a rise and fall of temperature inside. It depends on the circumstances. It may rise or temperature go down. So in that case, it will regulate, keep the temperature constant. So that is just in brief. If the temperature of the surroundings is low, that is cold. If you feel cold now, our surroundings is cold weather, so usually feel chilly. So we lose heat, and the body loses heat. But this will prevent, that is more loss of heat. What is the one? The air and this, what is that? That is the hypodermis, that is adipose tissue, will prevent the flow of heat from the body into the surroundings. How it happens? Here usually the thermal insulator, as you know, is a very good thermal insulator. It can insulate the body and avoid or prevent the loss of what is the flow of heat radiations from the body into surroundings. That is the one way. And what is the purpose of the air on this integumentary system? Here there is a mechanism is there by which the air sometimes what is that? It will pull upright. There is some muscular action is there. I don't want to go in detail. That is auto sensory. Sensor is there. It works on the auto sense. When the body feel cold, when the body temperature, because what is that? When the body losing more temperature, it will prevent what is that? There is one muscle is there. It can pull. What is that? Upright. So when the hairs lie upright to the skin, that will prevent loss of heat. That will obstruct the flow of heat from the body into surroundings. That is the kind of our hairs. No, that is why goosebump is there. Usually when the animals are, if you know in cold season, we see that hairs are that is pulling up upright. So that will prevent the loss of heat. That is the purpose of these hairs. Not only these hairs, the hairs present in our nose, ears and ears, no. That is other functions. They prevent entry of any dust particle or other particles into sensitive organs. That is through hairs and this. That is the purpose of this hair. So they have their own for integumentary system. That is in the body skin. That is a kind of that is thermoregulator. And if the temperature is cold, that is cold weather. If the temperature of the body rises, it is because of hot weather outside surroundings or any workout in the body, exercise in the body, that increases the temperature, body temperature. That will increase the body temperature. If that happens, increases, body should lose or cool down. That is one way is that these one is that in a relaxing position. At that time, sensors now these hairs are in the relaxing positions. They lie flat on the skin. That is, So if the light plasma will not obstruct the flow of heat, release the heat easily from the body into that. So with the light, what is that? Flat on the skin, heat can flow easily from the body into the surroundings. That is the purpose of this. And not only that, it will sense, no, this is a kind of eccrine sweat, more sweat, less salt. So it will sweat, no, what will come out from the duct, and will sense, no, synthesize water and salt, that is the cells are there, and release water on the surface, spread on the skin and evaporates. This kind of evaporation cool the upper surface, cool the body overall, cooling effect. Okay, what is that? So this is how we can control the body temperature. That is mainly this adipose, a layer of adipose tissue, a thermoregulator, this air integument system, and this gland actually a thermoregulator in the system. So that is a brief about this layer and this layer. Now come to the point, very important is that the topmost layer, epidermis. What is epidermis? Epidermis is very more important because I need to talk about the properties of these most important properties of these protein molecules. This plays an important role. Here again, epidermis layer contains, when you look into that structure, it has 5 different layers. Got that? Topmost layer. I will call this as different strata. Different strata of layers. Yeah, 5. A thin layer contains 5 different layers. It is called stratum. That is singular stratum. Stratum is plural. Carnium, stratum corneum. Just a typical biological or medical term. Stratum lucidum. Second one. Lucidum. Granulosum. Spinosum. Basale. Stratum corneum. Stratum lucidum. Stratum granulosum. Stratum spinosum. Stratum basale. That is the basal layer, top layer. What is that? Very important. Their cellular activity is very interesting, it's peculiar. So you don't see any other organs. That is a kind of cellular activity. Let me talk about what are the cells present in different limb. There is a bottom layer, basal layer, called stratum basale. Here, stem cells are residing. Usually, stem cells, there are stem cells are in adult stage also. There are different stem cells in different organs for regeneration, but they are not totipotent. So here the stem cell is unipotent. That stem cell is unipotent means this cell proliferate or what is that reproduce or cell undergo division, mitotic division, synthesize, make more and more cells. And here stem cell differentiate is only one type of cell, not many. So totipotent means that is usually seen in the embryonic, it is in embryonic state. They are the stem cell, you can differentiate into any organ or any cell. That is why embryonic state the different organs are developing. But if you say come across totipotent, it is unipotent and also multipotent. What is multipotent? A cell, stem cell can differentiate into 3 or 4 or 5 different cells. These are some organs we require now. Not only one cell is there, only one type of cell is producing. So the stem cell can produce 3 or 4 different cells in that organs. That is called multipotent. So organs we have in the adult stage also we have some stem cells are residing for the regeneration of the organs. So you listen, that is the one we require. Here a stem cell is unipotent. It is dividing constantly. Important is what? This epidermis, what is that? Epidermis is keep renewing. That is one. Regeneration and degeneration. Continuous. Regeneration and degeneration. And regeneration is faster in babies. That is new skin formation. Here the skin now, what is that? This there. What is that? Layer and layer now. It is moving up. The cells moving up and more regeneration. So this regenerated and what is that? Here constantly reproducing the cells, new cells. They are called keratinocytes. Afterwards, differentiating the keratinocytes. I'll tell you what is keratinocytes. And moving up, what is that? Finally, keratinocytes, more and more differentiation, and here sheds, constantly shedding from the skin as a scales. So keep shedding and keep reproducing. That is constant process throughout the life. But different rate at different ages. In babies, about 15 days, renewal of skin, that is, epidermis is renewed every 15 days, more or less. In younger, that is childhood stage, it is about 30 days renewal of skin, dermis. And after age, no, that is as the age, no, it may becomes what is that in 50 years, 1.5 months. After 50, it may take 2 months. Renewal of what is that? New generation of epidermis. It is generated and degenerated. It is generated and shed from the upper surface as a scale. So that is regeneration and degeneration is always taking place. Rate of that is different at different age. At baby stage, no, at early age we have faster. As with age, what is that? The rate of synthesis or rate of renewal, it decreases. Let us see what is that activities going on. No, we'll go in these different layers. In this layer, basal, here the cells residing are stem cells in the cuboidal state. And this cell is also there, Merkel cell, Merkel cells. So these cells, and one more point I will think about. The systemic capillary here extend up to this layer. We will not go into this epidermis. Blood capillaries will stop here, will not go behind this systemic capillaries, and nerve ending also here end with here. Systemic capillaries will not enter into this, or nerves will not enter, but nerve endings is little bit go in deeper, little into that. And nerve ending and blood vessels end up to this point only, flow is there. Behind this there is no capillary system or nerves in this system, no fiber in the system. So these Merkel cells are the, what is that, sensory cells, it is there. So 95% here, these cells are keratin cells, becomes keratinocytes. About 5% or 2 or 3%, what is that? These are Merkel cells. This purpose of these sensory organs, because here nerves are ending, but nerve endings are in touch with the surface. That is why Merkel cells. They can sense any they can sense any touch or anything from the environment. It can have the sense because it can extend this what is sensory sensation up to this level. So these are the ones it can sense from the surroundings, purpose of these Merkel cells. And here these are the cells, what is that, reproduce into, what is that, differentiate or proliferate or synthesize constantly. And as they synthesize, they will not reside over, they keep moving up, pushing up. As they push up, they differentiate more and more. That is the cellular activity, what is that? It's keep going and it's changing, what is that? It's differentiating more and more as moving up and up. It becomes, all of them becomes keratinocytes. What is keratinocytes? Here, this cell produces, what is the synthesize more and more keratin molecules. What is the keratin molecule? That is I am going to stress on. These intermediate filament are the keratin molecules. These are the intermediate filaments. So this cell is differentiated to synthesize this kind of protein, because this kind of protein molecules we need to require as perfect, what is that? As a to act as a barrier, uppermost layer. So here the cell synthesizes more and more keratin, more and more intermediate filament like this. Yeah. And during this pushing up from lower to upper, we see that there is always a change in the cellular shape and cellular activity and more differentiation, but keep moving up always. Moving up. So as they move, the shape changes like this spinosome, because you have spikes like this, spin like this, no spin or pickle like cell. What is that? That is to keep, what is that, interact or keep the cells closer and closer. Here tissue matter is more. Tissue matter, what is our tissue space decreases, cellular space increases, cell becomes closer and closer. Keep the cell tight junction, keep associating. That is why the spicer producers keep in association with the cell. Cells come and fuse together. That is why keeping the junction, tight junction. As they move up, what is that? Change like a diamond like. And in addition to that, more and more differentiation. Year to year, more differentiation, cellular shape. And as they move up, what is that? The three-dimensional structure becomes what now? Three-dimensional become two-dimensional, become more and more flat like this. So this cuboid becomes this shape, and becomes more flat, and this becomes more flat. And here the flat becomes almost what is that? Completely flat, thin flat like this, and completely tight junction. They come close, reduce the cell. So this space, 95% space occupied by the cells, only 5%. The tissue matter decreases when it comes to there is a changes will take place. Tissue matter decreases and cellular matter increases. That is a kind of differentiation. And also the cell make more and more keratin molecules. Keratin synthesis increases, and also one more, as the cell move up, it losing its organelles, including there is a nucleus and cytoplasm. So cytoplasmic content decreases, almost nil. So no nucleus disappear, organelles what is complete, almost filled with what is that? Keratin molecules. The cell is filled with keratin molecules. By this time, almost the cell is die, because no nucleus, cell is die. So these two layers, the cell is completely die. Here partially die, still some nucleus is there, still keratinization takes place, still with granules of keratin molecules. So it becomes changes again, more and more flat. Waves come down. These cells are complete dead, and complete what is that? Gives a tight junction, cellular junction, and contains, these cells contains only the intermediate filament is called keratin. It's all completely filled with keratin molecules, keratin fibers. Cell tight, no tissue space, because if tissue space is there, because there is a possibility of entry of what is that? Molecules from the surroundings into the body. Tissue is what is that? It can allow to diffuse even lipid molecules or gas molecules or some other. It can allow all the molecules of the air, that is we don't want all the content of the air contaminated, or water contaminated, microbes also enter. So that is the one kind of molecules. What kind of molecules? All cells are dead by the time. All organelles are becomes nil. All organelles disappear completely, vanishes completely. Your nucleus, no nucleus. It is completely filled with only these kind of fibrous keratin molecules. That is the layer, topmost, corneum layer. And this is the one we require, topmost layer is a chemically inert, because these molecules or layer or fibers are chemically inert. They will not allow, what is that? That is, the layer cannot attack by usually any chemicals except they are highly reactive. A strong acid or strong base can act on the skin, but usually a mild chemicals or any environment, what is there, any contamination, contaminated molecules or water contamination, or even microbes cannot attack these molecules, the fibrous molecules. That is a kind of inertness, that is a weak unit. So microbes cannot make entry. Microbes can make entry when there is a cut or accidents. So when they enter there, then they look after this. That is, we have immune cells are there, they look after to destroy the microbes here only almost. So that is a kind of layer which will protect now as a perfect barrier for no entry of water and no entry of any other molecules, because you are in touch with the atmospheric air, you are in touch with water also sometimes, any contamination, any other molecules, chemically inert, and not allow any molecules to entry, either this way or this way, a perfect barrier. That is the one we needed for this plant. And these, what is that, keep shedding new cells, because if they remain same, they will not protect. They will reproduce, synthesize, regenerate, and push up, and this kind of differentiation, more and more differentiated. The cells are more differentiated into this kind of cell, into this kind of cell, and this kind of cell. And finally, it will shed from the skin as scales. So that is the one we use that regeneration and degeneration is a must for the skin to keep our skin barrier. What is that? Perfectly a barrier protection against the all environmental contaminants, even microbes, anything. So that is the importance of this epidermis. That is the one. These are the molecules play an important role in protecting. What is that? These are the molecules, a cell containing these keratin molecules. A protector for our body. So that is one. And also I need to talk about some more about some other cells sometime. This keratin is called, they are keratinocytes. It is called this kind of moving up, no, this kind of process, what is that? More and more keratin molecules producing is called as keratinization. The process is called as keratinization as the molecules push up from the bottom to the surface. And keratin, this is the kind of differentiation, what is that? And in addition, we have Langerhans cells. Here, Langerhans cells are there. That is, immune cells are also here. Here also, more immune cells are there. These immune cells are there. These immune cells will act immediately if there is any, what is that, entry of microbe, accidental. If there is any break, there is a severe cut or any severe chemical action, there is a possibility of entry of microbes. These will look after this. And one more important note is that this is a barrier only for chemicals, but is not a barrier for UV radiations. It is a barrier, no? What is the UV radiations? UV radiations are the dangerous radiations. So now I want to brief about the radiations physics in brief. That is electromagnetic radiation. Now different radiation, you are calling them different micro electromagnetic radiations. Radiations you are calling on the basis of that is wavelength. You are learning about X-ray, gamma ray, X-ray, UV, visible, IR, infrared radiations, microwave, radio waves. All of them have their own applications. This in terms of wavelength, I am calling it as, it is in the range of 0.001 nanometer, more or less, approximately 0.01 nanometer range. Nanometer is what 10 to the power of 0.01 nanometer range. This is 0.1 nanometer range. This UV radiation between 100 and 400 nanometer. Visible radiation 400 to 700 nanometer. IR 700 to 10,000 nanometer. Microwave in centimeters. It is in meters and kilometers. Wavelength. What is wavelength? Radiation, we call this is a wave. Always electromagnetic radiation always transmit or it will transmit in the space in the form of wave. It is called one wave. Rest and break. This is wavelength. So this wavelength will differentiate these radiations. Wavelength, and as you know, these waves are like this. Not if the number of waves like this passing through a point in 1 second, it is called as one photon of radiation. That is, they always a molecule or atom usually absorb and emit this radiation in terms of photon. One molecule can absorb one photon at a time, or one molecule can emit, or one atom can emit one photon at a time. That is, number of waves passing through a point 1 second. It is called as one. That is about millions or billions of photons, because traveling at the speed of light, these waves are passing. And this wave number length, what is that? Defined frequency or wavelength. Is frequency or wavelength defines the energy of the radiation. Lesser the wavelength, more is energetic. Higher wavelength, less is energetic. On the basis of this, you are classifying this. Look at that, less energetic, little more energetic, and more, there is wavelength range decreases. Meter, kilometer into centimeter wavelength. They have their own, that is, they can travel with the speed of light only, because we are using for this communication, microwave radiation. This sending signals always throughout all the world, in also space also can send, because radiation can pass through any space. IR radiation, what is IR radiation? Heat radiations, temperature. The wavelengths between these nanometers. Visible, the light what you see, the light enter into eye and sense the object, that is visible radiation. Between wavelength between 400 to 700 nanometer. UV require 100 to 400 nanometer. That is energetic radiations. I call this, these are the dangerous radiations. And X-ray, because 0.1, we are using for, of course, X-ray of this bone structure. But immediately what is that? Only fraction of second the expose or removed. But you not keep exposing it only for a time. So we do not take more X-rays all the time. Dangerous. And gamma ray, it is even less, because this is the radiation coming from the nuclear atom bomb. Is more dangerous, because I am talking about this UV radiation. This UV radiation, X-ray radiation, gamma radiation can break the chemical bond. That is, these waves are energetic. So when they can, what is the penetrate any material? They can penetrate any biological system, penetrate even rock also. That kind of, what is that? Strength they have. Penetrate and break the chemical bond. So when they break the chemical bond, the molecules broken into pieces, make and break. Even DNA, make and break, pieces. And unwanted biological activities. DNA into what is that? Change that, yeah. The DNA molecule, what is that? Broken into pieces, again rejoining. Again biological rejoin that. Then gene changes, keep on changing. And change of gene will cause what is that? Unwanted biological synthesis, or what is that? Cancer it leads. That is, the UV radiation is the one which causes that, even X-ray, that gamma ray. And UV radiations, I am talking about the UV radiations. These UV radiations, there are different ranges are there. Nanometer 100 to 280 nanometer, it is quite energetic. These radiations, this range, all these radiations almost completely eliminated or removed in the upper atmosphere. That is in the ozone layer, ozone formation, ozone disappear. This will eliminate all these radiations. But still we have 280 to 320 nanometer, they can reach the Earth's surface. Even 320 to 400 nanometers. Not that energy, but this also is still energetic. They can penetrate, because source. Sun is the source of all the, what is that? Sun is the source of all this radiation. Of course, gamma is not there. Gamma is common, but sometimes extra. This UV visible IR and some microwave radiation also produce what is that? UV visible IR is the main source of sun. What is sun emitting? Visible light we can see. Sun is the one emitting all mixture of all these radiations, reach the surface of the earth. Heat radiations keep the earth atmosphere warm, visible to see. And UV radiation, that is one dangerous radiations. This also reach the surface of the earth. When these radiations reach the surface and when exposed to sun, what happens? They can penetrate. These are the one, penetrate. And this penetration of cell, what is that? What is that? They can sometimes, what is that? They break the macromolecules, break the chemical bond. Macromolecules, what is that? Fragmentation. Break into pieces. But we have a natural defense against this. That is what I want to talk about. This spinosome also contains, what is that? Other cell. That is, this basal layer also contains one more cell, melanocytes. Here, this basal layer contains melanocytes, and also some melanocytes are also over there. These melanocytes are the cell, a type of cells. These are cell contains what is that, some components are there. Some what is that some components. So that is component is called melanin pigment is there. So these melanocytes, the melanin pigment which can absorb this radiation, that is a photocell. It actually is a photocell mechanism is there in this cell. That photocell can absorb this radiation. What is that? The UV radiation in this range, absorb and change that what is that melanocyte into black melanin color. Melanin color is melanin pigment. So the melanin pigment is released in the form of black color when you touch with the UV radiations. So that pigment will come out or diffuses from the melanocytes and distributed all over. That is a kind of diffusion is there because the coloring material, that is black color. So only the melanin, actually it is a red or pinkish color initially. It will change into red color, that is black color when touched with UV radiation. So UV radiation penetrate when they touch with melanin. So melanocytes are the cells which can absorb. Melanin is absorbed and becomes a black color in change. That is why that UV radiation is removed or absorbed and melanin is distributed all over. It diffuses and distributed to all these colors. But the color is that your colors, if you are exposed to more sun rays, UV rays, intensity is there, the color becomes black because of pigmentation, melanin pigmentation. Melanin, it becomes a black pigment when exposed to UV radiations. More UV radiations, where the area, actually the areas, countries like our Indian subcontinent and African subcontinent, we exposed to more UV radiations compared to Western countries where there are no sunlight, no intensity of UV rays is very less. That is why usually the skin, what is that, melanin pigment is pink or red. That is why red color that is spread into all this skin is a red color. They look like red, but we look like black because it is a protection against UV radiations. And this melanin, what is diffused and will be concentrated in all the keratine cells. And presence of this black melanin here further prevent the entry of UV radiation. That is a mechanism. So the melanin pigment or melanin pigment, when the light or UV radiations fall on this, what is that, will scatter, will deviate the UV radiation, will not allow to penetrate into the skin, will just scatter it, deviate the change. The purpose of this melanin, what is that molecule, black melanin, what is thus produced in melanocytes, is spread all over the cells, filled with all over the all cells, and that melanin color will keep what is that scattering UV radiations. All this range will scatter out, not taking that. That is protecting our skin from this kind of mechanism. There is natural protection. That is why you look like black. But in Western countries, what is that? They are look black because intensity of UV light itself is not intense. Therefore, UV radiations are not intense. That is why the melanin color, what is a red and pink, will be distributed and becomes white and red or pinkish like this. So that is the reason the better in this area our skin should be what is that, a black color, protection everything. Because the skin is still it is white, so it will cause a problem like what is a skin cancer like this. They can enter UV radiation. Cause what is that, these molecules break, molecules of this break, even DNA molecule break and becomes what is that, cancerous cell. That is the reason. So melanin plays an important role protecting our skin, our body and our organ from these harmful radiations. And even more harmful. That is why this is even more what is that, if you take this body or what is that, very quickly all molecules become fragmentation. That is why nuclear radiations, not only when you put atom bomb now, not only the temperature destruct, in addition to that, this will destruct our biological system. That is even natural, any material will become what is not unwanted material, decompose. That is why that is atom bomb is the one. It is destructive. It's totally destructive. No? It destructs the entire system with this radiation as well as heat producing very high temperature. So that is the dangerous of the nuclear explosion. It is coming from the nuclear, that is nucleus of atoms. You are keep opening the nucleus of all atoms. Atom bomb is a principle on which it is based. So that is, I am talking about this. The purpose, what is that? Applications of these molecules. That is in this organ, skin organ. Let me take up the application of these molecules as well as in other organs like vascular system in the next class.